Hello, thank you for joining. Today I'm going to talk to you about RO-Crate, capturing fair research outputs in bioinformatics and beyond. My name is Phil Reed. I am a research community and training manager at the eScience Lab at the University of Manchester in the UK. I would like to extend the credits for producing these slides to my colleagues Eli Chadwick and Stian Solian-Reyes. First, a big question. What is RO-Crate? In short, RO-Crate is an open, community-driven, lightweight approach to packaging research objects with their metadata. Why should you be interested? Because using RO-Crate makes research outputs more attributable, interconnected, and fair. As I said, RO-Crate is an approach to packaging research data with their metadata. When we do research, we generate lots of outputs like data, workflows, publications, and so on. But these might all be published in different places. RO-Crate is a way of connecting the different parts of your research together and describing them with metadata, making your research more executable, discoverable, and reproducible. It does this using the power of linked data and RDF. It's about provenance, capturing the who, what, where, how of your research. There are many benefits of RO-Crates. RO-Crate uses linked data, which means RO-Crate metadata is naturally interconnected, interoperable, and queryable. The RO-Crate specification is general enough to be used in many different infrastructures and domains, and to describe all kinds of research objects. Custom RO-Crate profiles extend the specifications to meet the needs of a particular domain or community. RO-Crate extends and enables credit and attribution for all contributors, including those in currently under-recognized roles such as sample gathering, processing, and sample distribution. Here are some examples of RO-Crates in practice across bioinformatics. Elixir, the European Life Sciences Research Infrastructure, has multiple projects which publish pipeline executions as workflow run crates, capturing provenance such as who ran the pipeline, its inputs and outputs, and its execution environment. BioDT, Biodiversity Digital Twin, was an EU project to develop prototype digital twins and simulations in order to study biodiversity dynamics. We worked most closely with the group running simulations on wild crop relatives. They published the workflow executions as workflow run crates, which are then published on Destination Earth Data Lake. Galaxy and WiFX are also used in several other EU projects to publish workflow executions as workflow run crates. The Data Plant Consortium captures plant science experiments as annotated research contexts, ARC, complex objects based on the RO-Crate, which describe workflows, workflow executions, inputs, and results. The Biodiversity Genomics Europe (BGE) project is defining conventions for biodiversity genomics metadata as an RO-Crate profile, which is currently in development. This new profile aims to capture every stage from sample collection and preservation through to genome assembly and analysis. The COPO platform is used to export genomic data as RO-Crates. RO-Crate is now used within many fields beyond bioinformatics, including agriculture, languages and cultural heritage, climate science, materials science, astronomy and climate, and wider life sciences and health. A notable example is BioFair, the next generation of UK infrastructure for biosciences. There are many ways to use RO-Crate. First, workflows. These are used for describing computational workflows registered with the Workflow Hub platform. RO-Crate export is supported by workflow engines including Galaxy, Nextflow, WiFX, and CWL. Second, data archiving. RO-Crate is suitable for long-term data archival due to its clearly defined specification and use of well-supported web technologies. It can be used with any type of data. You do not need to change your data structure. RO-Crate is integrated into platforms such as RO-Hub, FedMseek, Paradisic, and Dataverse. And third, TREs. 5safe's RO crate captures the provenance of workflow executions within trusted research environments, or TREs. The profile includes processes like sign-off, workflow execution, and disclosure control, without requiring sensitive data to be published. We are using 5safe's RO crates in projects such as HDR UK and EOSC Trust. This is my final slide. A few final points. First, the specification is open source with an Apache 2.0 license. RO crate is developed in the open on GitHub. Anyone can join our global community to discuss applications, profiles, and tools. Start by attending a beginner-friendly drop-in session. For Europe, these are the first Thursday of the month at 9 AM UK time and the third Wednesday of the month at 3 PM UK time. See the table for other parts of the world. Thank you to everyone named below, especially to Eli and Stian, and thank you for listening. Please see our poster, number A118, for more information and come to me with any questions. If I cannot answer them, I'll find someone who can.